This is AP Biology, Chapter 9. We're going to be focusing on an introduction to genetics overall. We're going to talk about the chromosome and Gregor Mendel and his role in how we understand genetics or inheritance overall. So, genetics basically dates back to some of the earliest and most ancient attempts at doing selective breeding or artificial selection when it came to farming. And this is back in the 1800s. One of the most famous people associated with genetics is Gregor Mendel. He was an Austrian monk, and he did a lot of research and work on pea plants. And what he did was he actually did experiments and he documented the mechanism of inheritance with his experimentation. And it wasn't until the 1950s, so almost 100 years later, that the actual structure of DNA, which we now know is responsible for our genetics, was actually discovered. So Gregor Mendel was definitely ahead of his time. Now, the DNA that I'm referring to is located on chromosomes, and chromosomes are made up of DNA and a protein called a histone protein. And the typical somatic human cell, and by somatic I mean the typical cell that's not involved in sexual reproduction, the sperm or the egg. So every other cell in the human body has 46 chromosomes. And those 46 chromosomes are actually 23 pairs. One pair is from your mother, one pair is from your father. Now, these 23 matching pairs, we call them homologous, or homo means same, so homologous chromosomes. And that's because they carry the same genetics, which is responsible for making the same proteins or showing us the same traits or characteristics. They're often found at the same spots on a chromosome. So the area where a gene is located on the chromosome, we call a locus. And we'll talk more about locus as we look at the development of an embryo over time. So here is a graphic representation of what a chromosome looks like. So here's how we represent the homologous pairs. So here's one chromosome, here's another. Combined, these represent the homologous pair. If you were to actually unravel one of these segments, it would look like this. If you were to actually look at this rope, you would see that it's in fact made up of the DNA strand wrapped around these histone proteins. And so here they're showing the DNA strand, histone proteins. You wrap them all together. They become increasingly compacted until what you get overall looks like the modern day chromosome or homologous pair that we see in karyotypes. What Mendel discovered as a result of the years he spent looking at pea plants were the basic 4 principles of heredity. And he actually identified 2 of the major laws that govern inheritance. And let's look at what he did, and then we can talk about what he found. And so what he would do, he would take 2 pea plants that have contrasting traits, and then he would crisscross them together. Now, the parents or the original plants that he would use in his experiments were true breeding, which means that they only had one type of gene. And we call that the pea generation. So what he would do is he would take off the pollen from the anther and filament, the stamen. And what he would do, he was cut away these anthers, the part of the plant that contains the male reproductive organs. And then he would brush on the pollen from whatever plant he wants to the stigma of another plant. So he would intentionally take pollen or male parts from one plant and put it onto the stigma of another plant which contains the female parts, thus creating a hybrid or a crisscross. And so here you can see he's removing the anthers from a purple flower, and then he's brushing it onto a white flower. So in essence, he's crossing a purple and a white. And then what he's doing is allowing them to grow up over time. And then he looks at what happens in the offspring. So in this case, when he crossed a purple flower with a white flower, all of the offspring that were a result of that all turned out to be purple. And so he did this experiment several times with several different results. Now, the idea is that his hybrid offspring, so the resulting offspring that came from crossing of the 2 parents, are called the F1. And in this case, the P generation or parents were the purple and the white flower. The ones he crossed them and allowed them to grow up, those are your F1. And in our example, they were all purple. So let's go back. Here's our parents, our P generation. Here's our F1 generation. Then what he did was he allowed the F1 generation to self-pollinate, to reproduce within themselves. And in doing that, he allowed those offspring to grow up, which we call the F2 generation. And when he did that, he got a ratio of 3 purple plants to 1 white plant. And so here's what he concluded based on these results, is that if the F1 plants, if the only color that showed up was purple, then he knew that the purple had to be dominant. It had to be the one that shows up the most. However, there was still that white flower color. We just couldn't see it in the F1 generation. But in the F2 generation, we could see the white flower in 25% of the cases. And so we're going to say that that trait is recessive. And so Mendel concluded that every single trait or gene in our body is controlled by 2 genes. And he renamed these 2 different types of traits alleles. And that some alleles or genes are dominant, so they're going to show up more often, while others are recessive. And so some alleles are not going to show up as often, and we'll talk about that. And it turns out that Mendel had a lot of time on his hands, and he did a lot of crisscrosses. And so what he did was he crossed purple and white, and as we saw, he got a 3-to-1 ratio of 3 purple to 1 white when he looked at where the flowers would grow, whether they were axial, so off the side, or terminal, which is at the end. What he found is that, again, the axial was 3 times and the terminal was 1. When he looked at seed color, yellow versus green, he found that, again, in the F2 generation, 3 times yellow dominant showed up and 1 time the recessive green showed up. He did the same thing with seed shapes, round versus wrinkled. Again, the dominant round showed up 3 times and the wrinkled showed up 1 time. And so what he found is that there's 4 basic concepts to explain what was happening in this results. Number 1 is that there's going to be different versions of a gene, and we're going to call those alleles. So the alleles account for the variation in an inherited characteristic. And so here on this same chromosome at the same locus is the gene for flower color. However, there's the allele for purple versus the allele for white. And so on homologous pairs of chromosomes, it's possible to have 2 different alleles for the same gene. And then every single characteristic has 2 alleles, and you get 1 from your mom and 1 from your dad. And so a genetic locus is actually represented twice inside of a cell where we only pass on 1. So we could pass on the purple trait or we could pass on the white trait. It's completely random. Now, number 3 thing that happened as a result of Mendel's experiments is that if you have 2 alleles, then 1 allele will be dominant, and that's the one that's going to show up or determine what you look like, where the other allele will be recessive, and that means that you're not going to see it. Now, you still have it, but you just don't see it. So in our F1 generation, our purple flower color was dominant. So every flower had a purple and a white allele, yet the only one that we see that shows up is the dominant purple. So our white is then recessive. Now the 4th one is a law of segregation. And this says that yes, you have 2 alleles for a characteristic. However, they're going to separate when you make a gamete, a sperm or an egg. And you're going to pass on either, let's say, purple or pass on white. Whichever one you pass on is completely random. And therefore, the offspring that are made can have completely different combinations of genes and therefore look completely different. It's how you can make 3 offspring that are purple and 1 that looks white. So in conclusion for this kind of introduction to genetics and Mendel's overall idea is that Mendel was looking at phenotype, or what you look like, to determine genotype, or what your genes are. And an organism's phenotype is its physical appearance, whereas its genotype is its genetic makeup. And so it's possible for an organism to have a genetic makeup that contains a purple allele and a white allele, but their phenotype or their physical appearance will say that they look only like the dominant trait or only like the purple allele. So we have to make sure that we can talk about the two things as separate entities and understand that genotype determines phenotype.